It's important to maintain the cost is lower, but the cost is lower has to be balanced with, have you got people to do the tech, have you got the support for your clinician to use it?
You need to be a nerd of it. You need to be a genomics nerd to be involved in this. And you need to have a lot of experience and a lot of training. To be a clinical scientist is not a trivial thing.
Welcome to another episode of the MGI Tech podcast. My name is João, and today I'll be covering for our talented regular host, Will Suter. As we celebrate World Cancer Day today, we're spotlighting the impacts of genomics in precision medicine for oncology. We'll cover the integration of genomic testing in clinical practice, the role of technology in targeted treatments, and the challenges facing healthcare providers in making these advancements accessible to all. We're delighted to be joined by not just one, but a panel of three fantastic guests. We'll have Dr. Cheng Boon, a consultant oncologist at the University Hospital of Derby and Burton, and simultaneously medical director for OncoDNA. We also have Dr. Gareth Gerrard, a clinical scientist and the scientific lead for cancer genomics at the Southeast Genomic Laboratory Hub, Guy's and St. Thomas's. Finally, we're also joined by Filips Oleskovs, a business development project manager at MGI, who will tell us how MGI sequencing technology is suited for cancer research. We'll focus on the importance of collaboration, the promise of next-generation sequencing, and how partnerships like the one between OncoDNA and MGI are reshaping patient outcomes. Stay tuned, and we hope you enjoy our chat. First of all, thank you for joining us today, and I would like to ask you a little bit about your background. And so let's maybe start with you, Cheng. If you could share a little bit about your background and your work and what led you to focus on precision medicine in oncology.
As an oncologist, my message is that there's no particular age group or patient type that are immune from the diagnosis of cancer. What this means is that anybody could get it. And in my clinical practice, I see patients, predominantly adults, but it could be all ages. They are pediatric, children's cancer, all the way to people who maybe in their 90s would come to us and ask for treatments. My role as an oncologist is to ensure that I am up to speed to what is technically available, which may or may not be funded in the healthcare system. I am also trying to do my best to use the best solutions, options, depending on the patient's physical fitness and their ability to tolerate treatments. What this usually means is we try to find the targeted best solutions for those patients who are eligible. And the aim is to have the least toxicity with the best efficacy for as long as possible. There are patients that I would treat in the post-surgical or potentially curative section where the outcomes are very good. So my message is also that we shouldn't be all negative, but predominantly the cancer types that I treat are not curative. So the aim shouldn't be to try and push for a cure, but should be to live with the cancer as best as possible for as long as possible. On that note, I am committed to personalized medicine. This has been something that's been promised for 20 to 30 years. And we have to try and make sure we select the right patients for the right treatment at the right time. And this sometimes includes doing tests that would not be routinely available even 5 years ago. Solid tumor testing, which we will talk further, including those tests offered by OncoDeep, by OncoDNA, can provide an insight into the particular weaknesses in specific cancer tumor cells. And if we have the correct mutation, we may be able to find the correct targeted treatment, which would be predicted to give you the best outcomes. Precision cancer care should be available widely and as early as possible in the patient journey to hopefully improve the outcomes. Thank you.
Thank you very much. That was a very comprehensive answer and really, really insightful. Let's go to you, Gareth. So as someone who provides genomic testing for oncologists like Cheng in their clinical practice, what message do you have for oncologists like Cheng that want to use genomic testing to guide their clinical decision-making, treatment, and care?
Yeah, good question. So there are, as Cheng mentioned in his preamble there, that precision medicine is kind of where we're at the moment, and there's been a big focus on this. And it's nice to see it coming to the solid tumors. I mean, my background for most of my career was in the hematological malignancies, particularly the chronic myeloid leukemia, where we had, the advent of the first targeted therapy, imatinib, which transformed the disease. And we also sort of pioneered all the molecular monitoring and, using molecular markers and genomic markers for diagnosis and prognosis and stratification. So from then, the targeted therapies started to come into play in solid tumors, particularly the EGFR inhibitors in non-small cell lung cancer and the BRAF inhibitors in melanoma. And it's expanded from there. If you look at any timeline over the last 10 years, the way that these targeted therapies and precision medicine has really come to the forefront of cancer management, we can see just how quickly that has expanded, how many new markers are coming on. So really now we're really thinking about genomics as being the center of the whole precision medicine strategy in oncology. And for two things, really, because obviously we want to maximize efficacy and minimize any deleterious effects. In all medical interventions come with some risk. You don't get anything for free, as it were. So we want to use these specifically designed inhibitors in only the right patient group. And you can have patient groups like, for example, non-small cell lung cancer, where it's not one driver. It's really, there's a whole myriad of drivers. The various different EGFR variants may respond differently to different inhibitors. You've got KRAS G12C, BRAF, and then you've got all the various fusions, ALK, ROS, RET. All of these will have their own different response spectrums to each of the various myriad of inhibitors that there are there. And I think it's easy if for people like me who sort of live this, eat it, drink it, it's our focus every single day. It can be complicated, but if you're coming to it fresh, if you're not really embedded in this constantly evolving and changing world of genomics and the associated targeted therapies, it can be quite difficult. And we recognize that through having dialogue with our clinical colleagues who are trying to make sense of this information that we will give out as a lab. So when we design how we feed back this information, our reports about what is really, really useful information, we do talk, we do this in collaboration with our clinical colleagues to make sure that they're getting the information they need for, first of all, the standard of care, as in, I've got this patient here, what do I need to do now? But also because we're using more and more things like the comprehensive genomic panel screening, particularly on DNA. So we're not just looking at one or two genes, we're looking at several hundred genes. In fact, the panel that we use now is looking at over 600 genes. So there may be other targets in there which are not standard of care, but may be very useful for then clinical trial options, compassionate care options, but also, diagnostic, theranostic. Oh, right, they've got, loss of TP53. They may, not have such a good outcome on this particular therapy, or they may relapse quicker, and therefore we might want to monitor them slightly more, often than we would with someone who didn't have that particular genomic change. So, part of my role in this is to make sense of this, look at the horizon, see what's coming in, what new innovations, what's coming in through nice recommendations here in the UK, what's getting licensed, and then prepping to make sure that whatever technology we have is fit for purpose for that, but also start thinking then about the clinical context. How are we going to use this? How are our clinical colleagues going to use this? How are we going to get maximum benefit for the patients involved? And then open that dialogue with the associated oncology, oncologists, pathologists, surgeons, whoever it is in charge of these patients, so that the information that we give them maximizes that benefit.
Thank you, Gareth. I think you just touched a bit more on my next question, but I would like to ask you both, Gareth and Cheng, what do you see as the most significant barriers to implementing precision oncology in clinical practice today?
In order to answer that question, I have to, first of all, describe what roles I play. And because, number one, as a consultant oncologist, my role is actually very limited. I can only deliver systemic treatment such as chemotherapy, targeted treatment, and more importantly, those treatments must be available, funded by whether it's the public sector or the private sector or the insurance or the self-pay. So your role and your opportunity to influence change is very limited. But because in my role as the medical director for OncoDNA, I do work with experts like Gareth and scientist team in the OncoDNA team. And I also work with the public sector, the NHS Genomics and scientist team. And that really is the challenge because there's only 24 hours in a day. There are thousands and thousands of patients coming through everywhere. Cancer is one of the biggest challenges that we will have. And as an oncologist, I need to think frontline. I have about maybe 10, 20, 30 minutes with one new patient consultation, and we have to talk about, the standard of care, which usually is still traditionally chemotherapy, systemic treatment. But then how do you integrate all that information from the genomics revolution that's happening in the lab? How do I bring that to the patient and ensure that we speak in a way that they can understand? If you tell somebody, let's have chemotherapy, they know what it would be. They have TV shows, you watch on Netflix, you lose your hair, you've got, some celebrities having chemotherapy. That's easy. When you're talking about targeted treatment, it's all about managing expectations. So the challenge we have is that the expectations is this high. They think that you will have a magic treatment, it will go away. Fantastic. The reality is only a small number of genes have been found to be influenceable. We call it the driver mutation with the suitable targeted treatment. Because that targeted treatment depends on two factors. One, we need to find the target driver mutation. And secondly, we need someone to create a drug that would be able to target that safely without causing side effects. Now, those are big challenges because of the time and the cost needed to do the research, find the target, and then to get the trial, to get the drugs. And that could take 5, 10, 20 years to get from, initial idea to treatment delivery. So for me, the biggest barrier is the lack of time. The oncologist just has limited headspace, time space, and you have too many variations. So personalizing or precision oncology means you are not following the protocol. You are intentionally spending time to try and target this treatment, and you could do that via MDTs. That means in the UK we have multidisciplinary team meeting with scientists like Gareth and other similar colleagues, and we sort of try and integrate that into what we traditionally do already, which is we have a surgeon, we've got radiologists, we've got oncologists, we've got nurses, we've got dietitians, we've got everybody. But how do we integrate all that? And I find that is the biggest challenge because we've had 50, 60 years of developing my field as a clinical oncologist. That is STAT exams. Everybody knows what we're supposed to know. As a radiologist, there's also STAT exams. You need to have all that. But there's no such thing as a precision genomics or precision oncologist clinician. So I think that will change in the next 5 to 10 years, and we may have a new field where instead of depending on oncologist or clinician or a medical doctor, we have a specialist who works between the treatment. Currently, we have geneticists, but they are more with diagnosing the problem, whereupon they're not really using those findings from the genomics testing for treatment. And I foresee maybe in future, they will be subspecializing. Neither the oncologist takes the role on, or the geneticists will come into our field and take that on. But I think that's the major challenge that will make things much easier.
Amazing. Thanks a lot, Cheng. I mean, it's really interesting to hear about your dual role and how you have to wear different hats. On one hand, doing the frontline work as an oncologist, but on the other hand, being a medical director for OncoDNA, so seeing things from the other side. So thank you. That's really, really interesting to have those two takes on things. So let's go to you, Gareth. What from the other side of things, from the genomic testing service provider, how do you see what are the top significant barriers to you and to implementing precision oncology in clinical practice today?
Yeah, well, it really comes down to resources, really, and resourcing, and that breaks down into many things. I mean, money, a lot of this is very expensive, and uh, it's a challenge for all healthcare systems. I mean, I'm personally a big fan of socialized healthcare, and I'm very proud to work for the NHS. Um, and it does amazing things with the resources it has. So we have to use the resources very carefully, though, because it's the same pot of money that goes all around, and then you have to balance, very expensive genomic tests against, the other things that changes people's lives, like, replacement hip operations, and you've got to get the right balance. And luckily, that's not my job. There's someone else that makes those decisions, but I have to try and feed that information up about what is the priorities. But also, we're also an aging population, not just in this country, but worldwide. And so cancer has become more and more and more because we're not dying of other things. We've all got to die of something, unfortunately. And cancer is going to become more and more of those things because it is associated with age, even though you can get cancer at any age. But so there's that. I mean, there's money, basically. It's expensive. And this is why we have bodies like NICE in the UK who look at both, it's got to be data driven. You can't just say, hey, we've invented this. It's brilliant. Everyone go and use it, because these things are very expensive. The targeted therapies, for example. And also the technology we use is coming down in price relatively. We're getting a lot more bang for our bucks, but there's still an awful lot of capital expenditure that's involved. And once you sort of invest in the platform, you want to get 5, 6, 7, 8 years out of it because they're so expensive. But yet the technology is moving so quickly that things become obsolete really, really quickly as well, because there's always something new coming along. So we're looking for that sort of price bout. And from a lab perspective, our sample numbers are always going up. But the turnaround times we need to do are always coming down. And for a lot of these targets now, particularly for those urgent cases where it's an upfront treatment, like again, non-small cell lung cancer. Non-small cell lung cancer is the it's 50% of our work basically, and it's the one you need to get that result out really, really quickly because it's not end of standard of care. It's the one you want to start the treatment, and we know that almost every week that goes by, if that patient doesn't start one of these treatments, it impacts their outcome. So our focus, if you can get non-small cell lung cancer right, then everything else kind of falls into place. We're producing an enormous amount of data now as well. It's not just one gene and then all the various variants. We used to be where you'd be looking at, say, BRAF, B600, it's a single gene, it's a single base pair change, design your assay around that. EGFR kind of changed that, and KRAS as well, because it's all, it's got your exon 19s, you've got exon 21s, you've got your exon 20s, you have to try and work out what those are, then you get the rare variants, you get the combination variants, you've got all these fusion genes, then you've got ELBB2 amplification. What does that do? MET amplification, MET exon 14. All these new things are coming on, and we're guided by the National Genomic Test Directory. So there's, we have again this dialogue about these new markers that are coming on. Most of them are driven by what isn't, has a nice recommendation, but also what might have, diagnostic, theranostic significance. So we need people who have the knowledge, the training, and also the passion, because there's so much information that it's not a passion of yours to know this. There's too much to know. You can't be dispassionate and say, oh, well, it's not a 9-to-5 job. You don't get up in the morning and go, all right, I'm going to go and do this stuff, because it's too much. The information will just knock you over. You need to be, you need to be a nerd of it. You need to be a genomics nerd to be involved in this. And you need to have a lot of experience and a lot of training. To be a clinical scientist is not a trivial thing. Most people who are getting into clinical scientists, they already have a master's degree, or a lot of them already have a PhD. And then they do another 3 years training. It's another master's level. And then you're just a baby clinical scientist then. And then you get this tsunami of information. You're involved to be expected to, manage the lab and interpret this data. And the data that's coming off is changing all the time. Every 6 months, there's an update to National Genomic Test Directory. There might be a new clinical indication. There might be a whole load of new, samples coming our way because they've changed the eligibility criteria for something. So you might have just had a trickle of certain samples coming through, and now you'll have like 200 a month, and this all needs to be get out the door. It all needs to be signed off. It all needs to be authorized. It all needs to make sure it's good. We need to be able to have all of these assays need to be clinically validated under ISO 15189 and assessed through UCAS. It's all accredited. It's all done to clinical standard. And this is a real challenge in a fast-moving field, with limited resources. And there's only so many people who can engage with this data and make sense of it and be able to communicate it back to our clinical colleagues in a way. It's a science and an art. Luckily, it's the sort of thing, because it's such a fascinating field and because there's so much potential to help patients, to help real people, that it draws exceptional people. And I'm very lucky to work with some exceptional colleagues who are really pushing the field forward, and I just just stand back and cheer them on, basically, because they're doing amazing work. And that's what it comes to. I think so. Yeah, and you were looking at the limits, and I always want to silver light anything, and I just turn this into a cheering match for my amazing colleagues who are transforming the field.
Thank you so much. Those were really beautiful answers. Starting from you, Gareth, the way you were talking about obviously the limitation of costs, but it's also really interesting to hear about your passion, and we're all in here for the genomics geekiness. Thanks for bringing that up. That's the reason why we're doing this podcast. It's to hear your stories and to hear about your passion in the field. And let's move on now a little bit into more of the equipment that you use. Gareth, I have a question here for you. Your laboratory uses the OncoDeep kit, and I would like to ask you, how does it help you provide genomic testing service to oncologists, and how does it help with the promise of targeted treatments and precision medicine?
Yeah, I mean, there are a lot of providers of the same sort of technology, these comprehensive genomic panels, particularly looking at DNA. We look at DNA and RNA. And we tend to have a large DNA panel and a large RNA panel. And what really sort of put the OncoDeep kit on the map for us was when HRD, so the homologous recombination deficiency test, was going to be brought in-house for the high-grade serous ovarian cancers for the use of olaparib and bevacizumab. And not an easy test to do, because again, it's not a single gene, it's a genomic signature. And it's technology that's been developed over many years and it's sort of pioneered by another technology provider. And we were then tasked with bringing it in when it became centrally commissioned through the NHS. And what made OncoDNA really stand out was it was a large comprehensive panel. It covered all the genes that we needed to look at on the National Genomic Test Directory, at least for solid tumors, which was our focus. And it also did HRD native, and you didn't have to run it as a separate test. It all sort of came out for free, as it were, And the bioinformatics was all there, because all the bioinformatics, they were initially a bioinformatics company, was there. So it was a case of you run the assay, you upload the data to the bioinformatics, the web, the cloud-hosted bioinformatics pipeline, and then you've got all the various genomic targets that we wanted to see, and HRD, rather than having said this. And it's just like, well, it sounds too good to be true. But then we started talking to them and found they were very, very easy people to talk to. And this is something that is really of their benefit. I come back to communication again. So it doesn't feel like we're just buying a kit from someone and using it. They feel like technology partners, and that's a real boon. I think all of the providers of technology sort of get that to some extent, but this really does feel like a partnership. I think maybe because they're a slightly smaller company than some of the big tech giants we've worked with in the past. It sort of made them stand out. And then they were like, so, well, what do you need from us? And we said, well, your platform is really good, but could we have this as well? And they go, yeah, sure, we'll go away and do it. And then 2 weeks later, they've done it and they implemented it, and it's like, okay, this is good. And basically, it does the basic stuff's really good. It's a really good genomics panel, covers everything we want there. They've made it very, very easy. Get the HRD. We also get things like the other sort of mutational signatures that we're interested in. MSI, microsatellite instability, very useful, particularly in colorectals and endometrials, but also it's a pan-cancer marker for immunotherapy, same as TMB, tumor mutational burden. And all of these metrics come out. They're just there on every single sample we put through. There's no extra cost. There's no extra analysis there. It's just there, and the information is provided. And it makes our job as people who are trying to find out this information and interpret it and report it back so much easier. So, yeah, it's been a very, very good collaboration so far. And it does everything we need at this point. So, yeah, that's pretty much it. It makes our life easier. And when we don't have very much time or resources, that's a very important thing. And yeah, it just works very, very well.
Excellent.
Thanks a lot, Gareth.
Can I come in with a little bit of context as well?
Of course.
So the initial use of the HRD, which is a homologous recombination deficiency panel, was because a drug was invented for, I think, ovarian cancer initially, was licensed, was approved, was used for ovarian cancer. And because it was patented, the cost of this drug was high. And so the regulators mandated you have to use a test that was approved for this drug. So that's called a companion diagnostic as FDA rule, and they started with that. So the issue is that the way the drug and the test was licensed or approved is that you have to have one for the other. And this doesn't make sense to me as an oncologist. If I do a full blood count, I go to any lab, the results are the same. Whereupon this drug was approved with one test and only one provider, and that means it's a natural monopoly. And that created a problem for the drug company. When the patent of the drug company was coming to its end, they realized if I were to pay this company, I don't know how much, X amount of money for this specific test for every single patient, and you must remember, not every patient had this HRD status, and so not everybody is going to get their drug. So the drug company said, I am not going to pay for this anymore. I've had enough. It's not making sense anymore. That's when the regulators in Europe, UK started saying, we need to find another way. This test is not economical, and if we keep doing it the same way, we cannot afford the drug. So what the regulators in Europe and UK say, well, this is a generic test. We don't need to pay brand name money for this test. And so there is now, OncoDNA has a kit that provides a validated HRD result. So what we're doing is, this is equivalent to a generic drug. This is a generic test. And because it's generic and the OncoDNA is a bigger panel, a more advanced test, you get bigger bang for the buck. You get the HRD for free, and you get all the other tests that wasn't previously available on the more expensive panel for free. So that is the other reason why this works. So it takes time to explain it to the regulator that you're getting a bigger bang for buck by using generic. So you could learn to speak the language that the economists understand, the patient understand, because if not, they'll say, why are you changing? The drug companies say, well, this is a approved drug by FDA, the insurers say, they don't understand that. But once you start treating nitty-gritty day-to-day, doing the calculations, working with your economists, with your payers, with politicians, it takes time. And again, it comes back to my topic. Where is the time to do all this? I'm an oncologist. I'm supposed to be in the front line. I'm not there to lobby people. But you have to understand the bigger picture. And as an oncologist, it is hard. The only reason I know all this is because I work with OncoDNA. I have to know some of this. But some of the decisions we make sometimes, when you look at it doesn't make sense, but it is what it is.
That's fascinating. Again, like that dual role comes into play here. You obviously have this big insight into the ecosystem of things, but also have to do your work on the front line. So I can imagine it's kind of like, how do you deal with these two realities at the same time, and these constraints at the same time? Actually, my next question is specifically for you, Cheng, about OncoDNA, and it follows. There we have a quote here from OncoDNA's website where it's mentioned, next generation sequencing had the power to revolutionize the diagnosis and treatment of cancers and rare diseases and bring the right treatment to the right patient at the right time. So our question is, could you explain how next generation sequencing technology helps in the diagnosis of cancers for our listeners?
Thank you. This is a very tricky question to answer. Not because it's technically hard to answer, but because it's about managing the expectations. When you say, has had the power to revolutionize diagnosis and treatment of cancers and rare diseases, well, that's true, because there are quite a few cancers that are very much depending on one or two driver mutations, but those cancer types are rare. The first one was actually the one that revolutionized the treatment, was a condition called chronic myeloid leukemia, CML. And this condition, this cancer, was not responsive to chemotherapy. None of the treatment worked. They did bone marrow transplant, they did all sorts of treatment. It didn't work very well. And obviously, this was a long time ago, and they did the research. They found there was a driver mutation, and there was a drug, tyrosine kinase inhibitor, an oral tablet, which targets this specific pathway, and it worked beautifully. So from a condition that does not respond to chemo, doesn't have very good treatment, terrible outcomes, to something that you live with essentially for a long, long time, and they go into long-term remission, and so on and so forth. Great. So that is, it works. The issue we have is that many of the common cancers that we treat may not necessarily have one or two driver mutations. Now, those conditions are still depending on using older drugs like chemotherapy, and also potentially newer drugs like immunotherapy, and sometimes in combination with surgery, earlier diagnosis, all that to improve outcomes. But the issue, what we have is rare cancers and rare diseases, such as pediatric cancers, brain tumors, things like biliary tract cancers. Those are the ones that have very poor outcomes. And when we say poor outcomes, their median survival are, for better or worse, less than a lot of other more common cancers. And it's hard for these patients because, again, it's a bit like the CML condition. It doesn't respond very well to chemotherapy. By the time we diagnose them, it's quite late. By the time you have a brain tumor, you have headaches, you've had it for a while. So it's very hard to treat something that you don't screen for, that is diagnosed late. And technically, that means you've had it for a long time. So even if you have one driver mutation, you suddenly have a lot more mutations. But we never routinely tested all these tumors for this kind of mutations. Now we are starting to do this. Now we have the capacity to do it. Now even in the NHS, they're doing pediatric cancer. They have access to genomic. They look for mutation. They look for clinical trials. So it's slow progression. So I don't want to say that, we will tomorrow cure every cancer. It doesn't work that way. But on the other hand, we're using this kind of technology better. If we can combine it with better scanning, better screening, there are technology that the NHS is trialing, seeing if there's a way of detecting cancer before you even have symptoms. Now, if you can combine that with, let's say, earlier genomic testing, maybe there will be a smaller group, what we call a subset of patients that will have a specific driver mutation that we may have pharma who has incentive to create a drug to target that. And there may be a subset of patients that the outcomes will be completely different. But obviously, in its proper time, you need the ability to have access to scans timely manner. There's no wait for that. There's no wait for the genomics. The surgery is good. So everything has to go right for us to make a big difference. And so I've been an oncologist long enough to know that when we say revolution, we only revolutionize the field that we're in. So in the past, we do one gene at a time. So if you take a biopsy, a small needle into the tumor, you get about 1 or 2 mils of tumor. So you imagine this is a very small sample. And the pathologist will cut little slices of one slice, and you do one gene at a time. And now with this OncoDNA, you can do hundreds of genes at the same time. It saves, the most important thing is time. You don't have time to cut 300 of a slice to do, because you don't have enough tissue anyways. You can't keep biopsying the poor patient. It's a needle into a brain. Doesn't sound great. So you want to get as little sample as possible. We have as much information as possible. Not to say that I will change the treatment. Maybe, maybe not. But at least it gives you a clearer understanding of how to attack this cancer. If you have a target, we attack the target. If we don't have the target, let's go for standard treatment, which is maybe chemo, maybe radiotherapy. Do your conventional thing until we have a better study, or if industry or pharma has a better drug, then go for the trial. So we are better at selecting patients. And I think that's the biggest difference, because it's in the mind. We used to think, if you have breast tumor, everybody is the same. You have lung cancer, everybody is the same. You have brain tumor, everybody. Now we're starting to, okay, here we got XYZ mutation like Gareth said, and I don't want to bore you because it keeps changing. What I say today, if you listen to me next year, it will change, because every time I go to a drug meeting, they've got new drugs coming out. It's like very hard as an oncologist had and the medical director had to try to keep up with pharma, because they are being driven by, obviously, a blockbuster drug. Whereupon our job is to balance two things. One is you want to treat the patient, and two is my hands are tied. I have to spend within the budget. That's like Gareth said, I get that. And whether I work in the public social services or a sector, or even insurance, they will limit your spend because, you can go to talk about how insurance is formed, but it's usually three legs. One leg is you want to have universal coverage. One is affordable premiums, and you don't want a risk spiral. Everybody has to, contribute to this, and therefore you have to limit our spend. So, there's no easy way to square that. Once you start going into that, then you've heard in the UK there was the end of life thing. How much is a life worth? If somebody say I don't want treatment, is that the right thing? Or am I supposed to be talking? So it becomes, how do we square the circle? And I think the only solution would be you have to have a pressure valve. This kind of test is cheap enough. If you can't do it in the public sector, maybe the private sector, or maybe industry funding, maybe the drug companies will help. So I think there will be a fudge because, like Gareth said, there's only so many labs you can build. There's only so much money. There's only so many Gareths you can create. It takes years to train one Gareth. So it's very hard. And I don't see where the skill set, where the next generation of Gareths are coming. Is very hard because, if you're just looking at money, it doesn't pay a lot to be a hardworking scientist. The incentive is not there. The incentive is instead in the pharma. If you're talking about cancer, pharma is the biggest incentive. They attract the best people because they pay the best. But is that the right thing? Because you still need people like Gareth and myself to deliver the treatment. If you don't have us, there is no pharma. So they forget that. You pay so much there, you've just doomed yourself because you have people who are de-skilled. They wouldn't know how to use your drug anyways.
Fantastic. Thank you. I wanted to also ask you how, in that effort to diagnose cancer, how important is selecting the right sequencing platform?
This is a question that is a bit like, how do you create the best services? Like, for example, if you have a CT department, how do you choose between brand A, B, and C, and D? And a lot of it comes back to who is the radiologist, who is your radiographers, can you work as a team, is it economical, do you have a service contract, what happens with after sales? It's because overall the capital spend, the upfront spend, is not the main determinant. Actually, it's how throughput can be. So if you have a very high throughput service where you get hundreds and thousands of services that you have to deliver to a time, then you want reliability, you want easy to recruit staff, easy to fix, no breakdowns, and good quality assessment, because those are important. Can you give what the future proofing? Let's say a new mutation is coming, and there's a new drug. Can you test for it? And can you test it in a way that we can do it quickly and turn around the results fast enough? And then how do we check the quality results? Have you got a chain of people? If we got many hospitals, I can compare the result. So other things that are logistically important. And ultimately, I've never said it, but obviously cost is important. Your payers, you'll sit in the panel and they'll count per test how much it costs, what's the services charged, what is the staffing levels, the time. Can you run the services 18 hours, 12 hours? Can it automate? So all that may help. And as you get more data, next thing you think is, can you take away the human? Can you put AI in? I don't know, because I foresee there's not enough. Like I say, time is the problem. You don't have enough time to do all this manually. You have to think about future-proofing your service. And I hope our host, MGI, which is I'm very gracious to come for, will have all this already, and they will work with their local team. And the answer is, work as a team will be your solution.
Amazing. Thank you. And we will actually hear from Filips a little bit more about MGI's take on how to tackle these challenges. But I have another question for you, Cheng, which is related to everything you just said. When undertaking new research, what do you look for in a sequencer product? I think you already mentioned a lot of these aspects, but is there anything particular that you would pinpoint?
So the way we do clinical research in oncology, it depends on how big your clinical research would be. So you have the basic sciences, so the preclinical stuff, that usually is done by the university. They don't need a lot of this. Some of it is in-house. They might even do 1 gene or 2 gene because they know their target. So that is probably not going to apply to me. For clinical research in oncology particularly, most of the study will be things that have already gone through phase 1, phase 2. So we know it's your target is what. We know what you need. and we're just going to prove that it's better or equal to the standard of care. So for those, you want a test that is reliable. That means the data is good enough that you don't have a lot of false positive, false negative, because every test will have that. Every time you have a false positive, false negative, it affects the results of your trial. It makes your drug looks bad. So if let's say I'm wearing my pharma hat, let's say I'm a farmer, I want to do a drug and that would approve the drug. You're spending millions and millions and millions. Obviously, when you're doing this kind of commercial trial, money isn't the problem. What the problem is how quick can you recruit? So anything that's slow, no good. Anything that is fast, highly accurate, reliable, because you want to do as quickly and as many patients as possible, because you're depending on this kind of mutation to be detected. The reality is, I say, this kind of mutation is like finding a needle in a haystack. It is hard to find. You may have to screen hundreds of patients to find 1 or 2 patients. Nowadays is that kind of 1 or 2% kind of level, of which you want it to be universally available. So what this means, especially in the public sector kind of healthcare environment like the NHS, is that you have to make sure that the labs are able to do it. Now, whether you want to do it yourself and build your own sequencing machine in every hospital, that doesn't make sense to me. What they want is to go to the NHS DNA labs and say, okay, can you do this test for me? I'll give you X amount of money. Money isn't the problem. We just want the test to be done. And if, let's say, they have a panel that's compatible to their machine, the infrastructure, then it's dealing with the logistics, because you have to get the hospital to send the sample in a timely manner for you to recruit into the study. And that is the major challenge, the time. And so to me, a sequencer product, as long as you're at scale, you got good quality, you're fast, I would think that would be the priority. Other things are outside the lab. Things like, how quick in your courier services. You send it by Royal Mail, there's a strike, you're not going to get your sample in time. Or if you don't prepare your sample in the correct test tube, or you didn't put ice or something, and the tumor tissue is damaged, then you wasted everybody's time. So it becomes very pressured, because somebody's paying a lot of money for you, and if you can't deliver, you will cut the trial. And so if you look at the overall trial, phase 3 trials in the UK, the numbers have fallen. And the numbers have fallen because we only have predominantly a public sector healthcare. So people realize with the wait, all the bad publicity, people are being treated in the corridors, the NHS capacity to do additional work like clinical trial are very limited. We are not investing. We're just trying to cut spending, which is, you can cut spending, but you're not going to create revenue. Because in other places like the States, yes, they have a lot of health spending, but they also have big pharma that are billions and billions worth. So it comes and goes. You can't just say, I want to cut spending without creating revenue. Because if you have a big market, a lot of health spending, ultimately somebody has to make the drugs. And usually the drugs are made in the biggest market because it's quicker turnaround time, because it's competitive. So that's why I think here in the UK, I think it will be something that will be compatible to what the DNA labs will have to be able to deliver. So the biggest determining step will be something that they can work with our DNA technology currently installed.
Excellent. Thank you, Cheng. Great. At this stage of the podcast, I would like to introduce our next guest, which is Filips, who is representing MGI Tech. And MGI is a provider of sequencers that can be used to enable the automation of the OncoDeep kit, for example. My question for you, Filips, is, first of all, welcome. And we're coincidentally talking on World Cancer Day. And as Gareth and Cheng have been mentioning, collaboration is key in this field, and no patient should feel like they have to fight cancer alone. So my question for you is, how is the partnership between OncoDNA and MGI helping oncologists and clinical scientists like Gareth and Cheng working in genomics laboratories and oncology clinics to deliver an innovative genomics service?
So first of all, I wanted to thank Gareth and Cheng for this very insightful conversation, and it is also very exciting for me to hear these things, because in MGI, I'm looking on everything at the angle of technology, and of course I understand the bigger purpose we're working for, and hearing it from the people on the front line is very inspiring. And for the MGI OncoDNA collaboration, I think it really aligns very much with everything which was said today. So we are really looking how to make diagnosis using next generation sequencing as affordable and as available to broad patient circle. In our collaboration with OncoDNA, we feel like we're really making the difference here. And this also perfectly aligns with one of the main goals of MGI, which is to democratize, I may say, access to sequencing technologies.
Amazing. Yes, I remember Gareth mentioning before the importance of equity of access. So it's great that MGI are looking into it. And actually, my next question for you, Filips, is what unique features of MGI's technology make it actually particularly suited for cancer research?
So I would say one of the core uniquenesses of our technology is that it is an open platform, and we're not talking only about sequencing, but also about the automation, which I think also was mentioned a couple of times today. So we're really looking at the whole solutions to deliver results in the most efficient, both from viewing from context of time, costs, from all the aspects. And at the same time, of course, the quality is paramount for precision medicine applications. And this is where position of MGI is also very strong, because our core technology relies on, let's say, it's called DNA nanoball sequencing, which basically reduces clonal errors, which doesn't allow for index hopping. So in the end, you're getting really high quality data in a very efficient way. And one last thing, I guess, also very important for the field is that most of our equipment is CIVDR labeled. And this is, of course, very important for medical applications. And when we look at our collaboration with OncoDNA, this is also highlights that we're offering the whole solution, the whole workflow, which is CIVDR labeled from the beginning until the end. And this represents, I believe, a very good case for precision medicine and clinical applications.
Excellent. My next question is for all three of you, actually, and we're going to go back to the landscape of precision oncology and have a bit of more of a look at that. So the question is, what trends and advancements do you foresee shaping the field of precision oncology in the next 5 to 10 years? Maybe let's start with you, Filips, since you were just talking about these things.
Yes. So while we were talking, I actually was writing down some points, and what I see a lot is time and costs, I would say. And well, I believe that when we look in the future, we should see what are the main bottlenecks at the moment. And this actually aligns very well with what we believe in MGI. So in the next 5-10 years, we see sequencing costs dropping even lower than they are now. Want to note that at the moment, MGI is offering the only company that can offer sub $100 genome for the most high throughput systems. And we see the cost efficiency going even up. And at the same time, the time of sequencing is very likely to be dropping further and further, thus ensuring higher speed of response for clinicians and improving hopefully outcomes of the patients. So these are 2 points that I see changing or improving in the next years.
Amazing, thank you. And Gareth, what do you reckon?
There's so many things because it's such a fast-moving field. I mean, from a technology point of view, as someone who's involved in having to make technology choices, the things that's driving us at the moment really is, one, is cost. It's always there. But speed, agility, scalability. So from a cancer oncology point of view, the turnaround times are coming down. In the UK, there's something called the Cancer Genomics Improvement Program, where we're bringing down the end-to-end time in which we're meant to feed back a validated report is going from 21 days, whatever it is, down to about 10 days, 14 days from the biopsy being taken, so about 10 days in our lab. So what we need to do then is use whatever technology we have in place very efficiently. So there are some technologies you can get enormous amount of sequencing out of it, and that means the price per base is very, very cheap. But you need to be running that machine flat out, full, and that means running a lot of samples, particularly if they're oncology samples, which means that the turnaround times are then delayed, because you might have to wait a week till you have enough samples to fill it. We don't want to do that, particularly since we're always limited for space, and automation can only do so many samples. So our point of view is we want a platform that we can use smaller runs and keep it running, but the economy of scale is kept across that sort of flexibility. Particularly with platform, a lot of platforms that you guys have several lanes on a flow cell and then several flow cells, which allows you then to put multiple runs on, so we can just have the smaller batches running and keep the machine running all the time with smaller batches. That's the sort of thing we're looking at now. I know that's something the MGI do, and there are other technology providers who look down a similar way. Also, the other things that are really driving, I think, in the next 5 years, liquid biopsies, not just ctDNA, which is obviously, and this is another way of looking at cancer DNA, which is shed into the bloodstream, and then we can just take a blood sample rather than having to biopsy the actual tumor, and then extract these tiny bits of quite fragmented DNA from the plasma of the blood, and then sequence that. And it has a lot of advantages. The disadvantage is, of course, again, cost, because you have to sequence much deeper. You need to do 10 times deeper or whatever. So there's a cost implement, trying to square that. But it does mean huge benefits for patients who then may not have to have invasive biopsies. They can just take a blood test along with every other blood test, and it means it doesn't have to go through the whole pathology, being turned into a block, the block being cut and assessed, and then being sent to the lab, and then try to extract DNA and RNA and other biomarkers ready from it. I think also then other forms of liquid biopsy, just using cytology samples. So again, not going down this solid, having to have everything embedded in paraffin. We can just use, say, for example, EBUS wash or other cytology samples for extracting DNA and RNA, which means that the technical thing of not having to formalin fix things means we get better quality nucleic acid, which brings me on to the next thing I think is going to be the future, which is long read sequencing. So a lot of the technology we've been using so far is what's short read sequencing, and there are a couple of technology providers, and everyone's getting involved in this now, which has a lot of advantages, particularly for rare diseases, but also in oncology, and then treating and breaking this barrier between things which are DNA, which are RNA. When we think about the normal driver mutations, we usually think about DNA. A lot of things, we're talking about CML, BCR-ABLs, the fusion gene, and in the solid tumors there aren't so many which are fusion gene driven, but we're sort of interested in these. We usually use RNA. And I think when we go down things like the long read sequencing, and they tend to be using nanopores, then we can just start sequencing nucleic acid, whether it's DNA and RNA, and then we can start then just picking out, the mutations and the fusion genes. And also, because of the way this technology works, then we're going into multi-omics. We think about the epigenomics, particularly DNA methylation, which is very useful for certain solid tumors at the moment when we're looking at, the way that certain genes are silenced this way, but also then particularly something that's being driven in the neurological tumors at the moment, which is using epigenetic patterns, the patterns of the methylation, in order to sort of pinpoint and characterize exactly what type of tumor that is, because they all look the same under the microscope, but they respond very differently to different, target therapies, and also, whether you need to cut it out completely, whether and what sort of other therapies you need to use then, these epigenetic classifiers, which comes to this. So I think that multi-omics. So again, this is probably going on 5 years, 6 years, 10 years time, but I think that's the way the technology is going. And there'll be something else new on the horizon, which we haven't quite worked out yet, but it'll come in, and there'll be another disruptive technology. We'll all jump on that probably in 7 or 8 years time. But I think that's the way things are going.
Thank you so much, Gareth. That was really fascinating. And Cheng, what do you think?
Thank you. I'm thinking with my oncologist hat. I'm partway through my career, and I've seen the first revolution where there was promise it's going to be personalized medicine, CML, like you say, APL, BCR, mutation looks great, fantastic outcomes. But then everything became slower. There's no more mutations to mine. All the easy targets have been found. So the cycle of expectations and, happy and not happy, that's part of being an oncologist. So I can see that we will have very good expectations. Everybody thinks and then suddenly we'll have a dip because there's no drug for a while, and then we find a new target. It's always like that. The second thing I need to be aware of is, how am I going to keep up with you guys? Philip and his $100 test. That means everybody will want one. The question is, how am I going to interpret it? Are you going to automate it and find a drug for me? Or I'm going to have to interpret every single patient? Because, for example, I can do a full blood count for 50 pounds or 30 pounds per patient. Every time they have a cycle of chemo, they may have 2 or 3 times they do that. If I were to do this with this technology, I need enough headspace to interpret it. For me to interpret a full blood count, it takes me 1 minute, 30 seconds. For me to interpret your report with 500-600 genes, how long does it take me? So we need to have a way to standardize it. And it may be that we create a specialism. What I see is professionalizing the field. Here we have too many technologies. You have to go through so many hoops, IVDA, all that. Yes, I get that. But what you're not seeing is that you don't have a medical person to interpret it. That means, like radiologists, I don't care whether you're brand ABC, MRI, or CT scan. When you have a radiologist interpret it, it all comes back to the same. As an oncologist, I want it to be that easy. I just want to press a button. I want to get a report. It tells me exactly what I need to do. I want it as simple. I know it's not possible because the field is changing. But at least having somebody there to take some of the mental headspace in, or if there's a new drug, I need someone to countersign it to persuade my regulators or my payers. So it really, having the technology is easy. Having the support, as like my role for OncoDNA, is a hard bit, because you need a trained oncologist, or at least somebody who's able to interpret what to do versus what my hands are tied. My oncology protocol says you have to have this. And so it's about bridging the gap. All great, all sci-fi. It's like talking to a radiologist. They have 5T or 3 Tesla or 5 Tesla magnets, MRI, fantastic, and see into the brain or the future. The question is, do I have the technology to treat that? And so, great that you are cute, you're all fantastic in the lab. I'm infantry. I don't have James Bond. I'm just infantry. I'm just trying to deliver what you tell me. So it's important to maintain the cost is lower, but the cost is lower has to be balanced with, have you got people to do the tech support? Have you got the support for your clinician to use it? Having the best drones in the world, when somebody doesn't know how to use it, it will cause more problem. You tell me there's a mutation, I use the drug wrongly, it causes more side effects, use up more funding, and that makes everybody disappointed. But I'm not negative, I'm positive. I do think that, in about 5-10 years, we will be using this routinely. We wouldn't need to talk to Gareth or Philip. All this will be automated, and it's human to human. That's when you go to any hospital, you don't even know what brand of scanner they use. I don't know, but it works in the background because the people that make that decision what to choose, it's not me. I don't want to involve the oncologist. There's not enough headspace. I don't have the time to do this. So that's what I see, to automate it completely, that I can just focus on doing my thing.
Thank you so much, Cheng. I think that's it's really fascinating to hear your analogy with the military, because it really helps us visualize how important it is, the support you need to meet the excessive demands that you have in your frontline work. So it's a brilliant analogy. We have a final question, which is related again to World Cancer Day. We're coincidentally speaking on this very important day for cancer research, and from this conversation, we have like some key takeaways that came from this chat. One, that patients are obviously the priority, and that no patient should have to fight cancer alone. The second one, as an example of this collaboration between OncoDNA and MGI Tech, that partnerships can help broker access to innovation. And that running a genomic service is definitely easier when you work together, when you work collaboratively. And so that these conversations are so important, and it's really important to share this with the public as well. So on that note, do you any of you three have any final comments for World Cancer Day and anything you'd like to share? Maybe let's start with you, Cheng.
Thank you. For World Cancer Day, I think we all agree that we need to make it easier for patients, but also for their clinicians to ask and to use this genomic testing meaningfully. We have to work together with our lab partners, such as experts like Gareth, Philip, make the science not just easy to understand, but completely reliable enough that you don't even need to know what the platform is. Because when you focus on the cancer, the diagnosis, you don't have time to worry about all that, how it works, what kind of sample. You just press a button, things work, just like you go for a CT scan, it's done. And the benefits of testing, especially when you test at scale and how you use this targeted treatment, will be transformational. It not just can be, it will be transformational to the right patient at the right time with the better health outcomes. The wrong way of doing this kind of genomic testing is we do it very late in their diagnosis, in their pathway. We're not well, we're poorly. Then in that case, you've just wasted everybody's money and time because you cannot make the change. But if you do it early enough for the right patient, you find the right patient, the right drug, it is transformational. And we have, from history, this is already happening. And I wish everybody a World Cancer Day, and we hope to make more progress year to year. But I hope a big bang to the outcomes, improve outcomes dramatically. But in my role, I have to manage that most likely will make incremental progress. Incremental, but when you add it all together, it's real progress, reliable, useful progress.
Amazing. Very, very inspiring. And Filips, do you have any comments for World Cancer Day?
Yeah, I just wanted to strongly agree with Cheng on his comments. So, as we mentioned before, the patient should not be fighting cancer alone, but also us as a service providers, clinicians, equipment providers, kit providers, and panel providers like OncoDNA. We are working together. We're all piece of one big picture. And this is by this synergy, we are actually able to enable the best service to our customers. And from MGI side, we have made our mission to provide fast, reliable, stable sequencing equipment and automation for the world. And we are certain that there is a lot of there are a lot of benefits for cancer patients in the end from synergies like the one we have with OncoDNA.
Thank you so much, Filips. And Gareth, do you have any final comments on World Cancer Day?
Absolutely. I think one thing that's really going to come into the fore is, again, so we talk about communication, is the information and the way we handle information and the way that people, particularly, there's so much information. I mean, so if you're battling cancer or you are supporting someone who has cancer, there is so much information out there, and that information can be overwhelming. So I think one of our roles is to try and there's an education role in everything we do as well, is to be able to empower people with the knowledge to be able to understand and feel they're part of the conversation about their treatment and what's best for them. And also the way that their healthcare systems prioritize what they want to do. And you can only, be involved in that if you have the understanding about what's going on. And I think there's going to be some big changes about the way we handle data, we look at data, because the amount of data that's being produced is enormous. And we only need a little bit of it, but there might still be lots of other useful information in that. Again, aging population, something we haven't mentioned is pharmacogenomics and the way that our own genomes affect the way that we metabolize and respond to different drugs, including chemotherapy and targeted therapies. And that's the sort of thing that's good to know earlier on. There's certain predisposition syndromes that make us more likely to have certain cancers, like Lynch syndrome or the BRCA1, BRCA2 genes. And there's a whole host of other sort of cancer predisposition genes there. You want to know those earlier on if you want to see what people's risks are, so that they can be managed more effectively, so you can screen these things more effectively. So the best thing for dealing with cancer is catching it early. And actually, It's our surgical colleagues who've got the best curative for most of the solid tumors, is to cut it out before it spreads elsewhere. And so a lot of the technology we're thinking about now is how can we detect cancer earlier and deal with it earlier. But the main thing is there's always hope. Technology is moving forward, and there are a huge number of people with a great passion for helping people involved in this. There's so many of my brilliant colleagues who've dedicated their lives really to trying to push this forward and trying to make people's lives better. And that's the main thing. I think, we're all unified in this, that we want to push this technology forward, push care forward, and make everyone as healthy and as fit as possible for as long as possible.
Well, what a beautiful and inspiring way to finish this conversation. Thank you, Gareth. But I would also like to thank all three of you for your beautiful insights and takes on this, and for your time, obviously, for being here and speaking with us. And for our listeners, thank you for listening, and feel free to share any comments you have for World Cancer Day and to follow the podcast on your favorite platforms. Thank you all for joining us. Thanks a lot.
Thank you very much.
Thank you.